So now we're going to go through each of the different parts individually. This is glycolysis. Don't worry, you don't have to know all of this. I'm actually going to make it bigger. But I just wanted to let you see the whole thing, so I made it small so you could see it all. So this is just to show you the glucose is going to get split in two. And I'm going to use this to show you that it has many different steps. Different organisms may have, or different species may have a different number of steps. Every step takes a different enzyme. And I am going to show you substrate phosphorylation in this picture. So I'm going to enlarge it for you. So here we start with glucose at the top. That has 6 carbons. And this first step, right here, we're actually going to be using ATP. So 2 molecules of ATP are actually going to be used. So 2 ATPs are used in the very first phase of glycolysis. And look, ATP, 3 phosphates, follow the arrow down, ADP. The reason why and only 2 phosphates is because look what happened. Phosphate one on that side, phosphate one on this side. So it's using 2 ATP. So over here on the left side that says minus 2 ATP. We still have 6 carbons. And right here it's going to get split into 2 carbons. 2 molecules each having 3 carbons. Notice they each have a phosphate. And in this next part, we're going to be making NADH. So here's NAD+, taking up electrons and hydrogens and energy. Same thing on the other side. So this is where we're making those 2 electron carriers. And this next part is going to show the substrate phosphorylation. So here we have this molecule right here. So there's a phosphate on it. Everybody see that? PGA with a phosphate. You have the ADP, 2 phosphates. Follow the arrow around. Now we have 3 phosphates. T means tri, which means 3. This phosphate came off PGAP. There it is. Comes off here, goes on to ADP and becomes ATP. Notice now we have PGA. See that? PGA. So PGA with the phosphate gives its phosphate to ADP. Now it's just PGA. Everybody see that? In the next step, we're actually going to be making some water here. We're going to be making some more ATPs through substrate phosphorylation. The phosphate comes off, goes on ADP, and becomes ATP. You do not need to know all these details. The main thing you need to know from this is that there's many steps. Each step has its own enzyme. You can know what's being produced. How many ATPs, how many NADPHs. What does it mean by substrate phosphorylation? So let's go through that substrate phosphorylation one more time. So glycolysis, splitting of the glucose. And we're going to focus on this part that's in yellow. You don't need to know any of these names, just the first name and the last name. So glucose and at the very end 2 pyruvate. Then you do not need to know any of the other names. So this is just illustrates substrate phosphorylation. So we have a molecule that has 3 carbons, another molecule that has 3 carbons, and they each have a phosphate. ADP, which has 2 phosphates, follow the arrow around, becomes a TP with 3 phosphates. Same thing on the other side. So here we have molecules, 3 carbons with a phosphate. We give a phosphate to ADP, and now we have 2 molecules of 3 carbons. So phosphorylation just means to make ATP. So it made ATP by taking off a phosphate from a substrate. It has nothing to do with oxidation and reduction. Nothing to do with chemiosmosis, anything like that. So following glycolysis, we come to the transition step, which is the link cycle, so oxidation of pyruvate. So that's very important. It's a key connector that's going to link glycolysis to the rest of cellular respiration. And it is going to take place in the matrix of the mitochondria. So at the end of glycolysis, we have 2 pyruvates. They still have a lot of energy in them. And pyruvate oxidation is going to get a lot of that energy out to be used to make ATP. So here's an illustration of the transition step. So 1 glucose is split into 2 pyruvates. Now notice this is also called pyruvic acid. So pyruvic acid and pyruvate are the same thing. And that is in the cytosol, or the cytoplasm. Now that is going to, these 2 pyruvates are going to be converted into something called acetyl-CoA. Carboxyl groups are going to be removed from each pyruvate, and they're going to get released as, look right here, 2 carbon dioxides. Everybody see that? So 2 molecules of carbon dioxide are going to get released. That's 2 out of the 6 that were originally present in the glucose. See that in the picture? Yeah, notice it's in the matrix. You had 3 carbons, so now a carbon leaves, so now it's only going to have 2 carbons. So the oxidized 2 carbon molecule in the acetyl group is going to be attached to something called coenzyme A. And it's going to form acetyl-CoA. So the most important thing for you is to know that in the transition the transition step, alright, transition step right here, these 2 molecules are going to go into the matrix. It's going to give off 2 carbon dioxides, and it's going to form acetyl-CoA, or coenzyme A. And that is what's going to be joining right here into the Krebs cycle. So acetyl-CoA is a transition molecule that now is going to enter the Krebs cycle. Another name for Krebs cycle is citric acid cycle. And this is a picture of the Krebs cycle. So once a pyruvate is converted into acetyl-CoA, or coenzyme A, now that is going to enter the Krebs cycle. And just like the Calvin cycle, it's going to go around 2 times. And every different step is going to be controlled by a different enzyme. Now let me show you why it's also called the citric acid cycle. You do not need to know any of these names except for that first one, citrate or citric acid. Citrate, also known as citric acid, is the first molecule that's formed. Okay, so this is the beginning of the citric acid cycle. Citric acid cycle because that is the name of the first molecule. You do not need to know all these things here. I'll tell you exactly what you need to know about the Krebs cycle. First of all, you need to know it's also called the citric acid cycle. Make sure you know where it is. transition step and the Krebs cycle are both in the matrix of the mitochondria. It's called the citric acid as well as the Krebs cycle because the first molecule that is formed is citric acid, also known as citrate. Let's just take a look at it. Let's just see what's being formed. Look, there's a carbon dioxide given off, carbon dioxide given off. Here it's making an NADH, here it's making an NADH, here it's making an ATP, here it's making FADH2, here it's making an NADH, goes around one more time. 2 NADHs, 2 more carbon dioxides given off, that's a total of 4 carbon dioxides. So the glucose is completely broken down now. ATP makes another FADH2 and another NADH. What you need to focus on, besides what I just said you needed to know, are the outputs. Okay, so let's go through again what you need to know about the Krebs cycle. Alright, this is the third stage, glycolysis, then the transition step. The transition molecule, acetyl-CoA, enters the Krebs cycle. Transition step and the Krebs cycle are both in the matrix of the mitochondria. Krebs cycle is also called citric acid cycle because the first molecule that is formed is citric acid. It goes around twice. The result of going around twice is this: 4 carbon dioxides, 2 ATPs, 6 NADHs, and 2 FADH2s. Don't worry about knowing that inputs, don't worry about knowing the net result. So again, what you need to know is it's a transition molecule that enters it, acetyl-CoA. First molecule that formed is called the citric acid or citrate, so it's called the citric acid cycle. It goes around twice. The result of going around twice are these outputs: 4 carbon dioxides. The glucose has now been completely broken down, completely oxidized. 2 ATPs through substrate phosphorylation. 6 NADHs, 2 FADH2s. These electron carriers, when they're made, go over to the electron transport system. So this is the Krebs cycle. So next we have the electron transport system. If you remember, when we filled out that chart, I said for every NADH we make how many ATPs? For every NADH, how many ATPs do we make? 3. How many ATPs do we make for every FADH2? We only make 2. Right, now I'm going to explain why. So both NADHs and FADH2s are electron carriers. They are carrying electrons to the electron transport system. And that energy that's released as electrons go from a high energy level to a lower energy level is going to be used to pump hydrogens, right? A little bit different, not into the thylakoid space, but they're going to be making a concentration gradient so that the hydrogens will flow through ATP synthase again and make ATP. So let's look at what I have in blue. Right here's NADH. It goes to the very first molecule. So NADH goes to the very first molecule. And notice in this process right here, it's making ATP. Let's just follow the NADH. Here again, it goes right here, makes another ATP. Here it's going to make another ATP. So NADH is making 1, 2, 3 ATPs. But look at FADH2. FADH2 skips. So skips this first step. So since it's skipping that first step, it's not going to be able to make this first ATP. So it's going to bring it over to the second molecule, not the first molecule. So it's not going to be able to make that first ATP. So it makes an ATP here, and it makes an ATP here. So it only makes 2 ATPs. Okay, so electron transport system has another name for it, also called the cytochrome system. So when you hear the word cytochrome system, that is the electron transport system. So for every NADH, we're going to make 3 ATPs. But for every FADH2, we're only going to make 2. And that is because FADH2 brings its electrons to the second step. It's going to skip the first step. So it's bringing its electrons further on in the cycle. So it only makes 2 ATPs. So these electron carriers are really, really, really important because their electrons are going to be used to make most of the ATP. Now let's look at the very last step, the very bottom of the electron transport system. And let's look what happens to the oxygen that you are breathing in. This is why you need oxygen. Oxygen right here is a final electron acceptor. It is going to get reduced, and it is going to become water. Pay attention. The oxygen that you breathe in is used as a final electron acceptor in the electron transport system. That oxygen gets reduced, gains electrons, hydrogens, and energy, and the oxygen you breathe in becomes water. Alright? So you are making water. So whenever you make ATP, you are making water. Alright, here's a little story for you. So my twin sister and I went on a lot of field trips for school. And one time, we went to Seminole Canyon. It's a state park in Texas. And this place, very, very, very few people seem to have gone. There wasn't anybody there. There wasn't any lights. There were some bathrooms, and there was some faucets. You turn the faucets, scorpions would come out, and there's no one there. So Rebecca and I and our classmates decided that we were going to go out into the desert, because it's in a desert. It was kind of scary. It was nighttime. We had our flashlights, and there's I don't know what you call it, herds of tarantulas or something. There's all these tarantulas in the desert, and they're trying to surround us. It was really weird. I didn't know tarantulas would do that. So we decided that we wanted to catch one. We happened to have a grocery bag with us, so we captured one and put it into a grocery bag, and then we put it in a cooler. We got back to the car. We brought it back, and when we got back to the school, called up my mom. My mom said, "No, you can't have a tarantula." Okay, so we didn't want to kill a tarantula. Well, we didn't want to smash it or anything. So I decided that I was going to use it as a paperweight. And our teacher was very helpful, trying all these different things, trying to kill it without crushing it. Tried all these different things. It didn't work. So finally he had the idea that he would try some cyanide, because there was some cyanide in the chemistry lab. So we had it in a glass jar, and he put a little cyanide in the jar, and within a minute it was dead. It's quite amazing. We still have the tarantula. Rebecca has the tarantula. So what happens, with poisons. Look what I have over here. Cyanide stopped the electrons. Here's cyanide right here. Stop the electrons from being passed on. So if there's no electron can be passed on right here, the whole thing is going to shut down. Can't get to ATP synthase. Hydrogens can't go through. So stop the electrons from being passed down the electron transport system. Everything shut down. Tarantula died. So aerobic respiration, we have 4 steps. Glycolysis. Glucose is converted to 2 pyruvates, making 2 ATPs. Pyruvate goes into the matrix of the mitochondria, gives off some carbon dioxide. Transition step is the formation of the acetyl coenzyme A. That transition molecule, acetyl-CoA, still in the matrix of the mitochondria, enters the citric acid cycle. 2 ATPs are made. Both these places here, they're made through substrate phosphorylation. The electron carriers that are made in each of these stages, pyruvate, going from glucose to pyruvate, makes 2 NADHs, transition step makes 2 NADHs, citric acid cycle going around twice, makes a lot of electron carriers, and they all go to the electron transport system where chemiosmosis takes place, and through chemiosmosis, oxidative phosphorylation in the cristae of the mitochondria makes 32 ATPs. So now let's look at chemiosmosis in aerobic respiration. So it's going to involve the electron transfer chain, just like in photosynthesis. However, in this case, it's going to be on the membranes of the cristae. Energy is going to get released. Electrons are passed on from one carrier to another. The energy is going to be used to pump hydrogen ions, in this case, into the intermembrane space. In photosynthesis, it goes in the thylakoid space. Here it's in the intermembrane space. Remember, the mitochondria has 2 membranes. Hydrogen ions are going to come from the matrix. They're going to diffuse back into the matrix through the channels of ATP synthase. And as the hydrogens go through ATP synthase, remember those little motors inside? That motion creates energy. And energy is used to turn the motors around inside ATP synthase. Every time that they turn around, make 3 ATPs. So that's oxidative phosphorylation. So let's look at that again. So let's look at the top part. Intermembrane space. Here's the matrix. There's the cristae. Intermitochondrial membrane. Now, this is on the cristae. All those different electron carriers here, you have NADH. They're going to be bringing electrons to the electron transport system. Drop off electrons, or see there, NAD becomes NADH. So NADH gets oxidized, releases the electrons, releases the hydrogens and the energy, and the electrons are going to go down the electron transport system, just like in photosynthesis. As the electrons go down the electron transport system, in this case, the hydrogens are going to get pumped on this part into the intermembrane space. Everybody see that? So the one place I can get to the other side is that, and that is through ATP synthase. So let's go to the bottom picture. So let's start right here. NADH, FADH2, electron carriers. NADH is made in glycolysis, transition step, and in the Krebs cycle. FADH2 is made in the Krebs cycle. So these are all electron carriers. Right? They are in the matrix. Right here. Look right here. Follow them. Release their electrons. Hydrogens also come off when NADH and FADH2 are oxidized. They're going to give up their electrons and their hydrogens. Hydrogens right here. As electrons go down the electron transport system, they're going to release energy to pump these hydrogens from this side into here, into the intermembrane space. So they're all going to be building up on this side. So they're creating a concentration gradient, meaning there's a lot on one side and there's a little bit on the other side. Just like in photosynthesis, the hydrogens are positive, they're repelling each other, and they want to reach equilibrium. Right here, they go through ATP synthase. That motion as they go through ATP synthase creates energy to enable the motors inside to spin around. As they spin around, they make a bond between ADP and P to make ATP. Okay, one more time. Electron carriers are super important for chemiosmosis because they're going to drop off electrons and hydrogens. Electrons go down the electron transport system in the cristae. As they go down, they're going to be releasing energy. Energy is going to be used to pump these hydrogens, which could have been released by the NADH and the FADH2 from the matrix through the cristae into the intermembrane space. And this time, let's follow the electron. So electron comes for NADH, FADH2, go down the electron transport system. Energy is used to pump the hydrogens inside the intermembrane space. Follow around. Oh, here it is. Gets picked up by the oxygen and becomes water. Right here at the end of the electron transport system. But all these hydrogens are building up, trying to get away from each other. Only one place they can make a break for it. ATP synthase. So hydrogens are streaming through here, create energy by their motion. That allows the motors inside to spin around, make a bond between ADP and P to create ATP. For every NADH, they're going to be making 3 ATPs. For every FADH2, they're going to be making 2 ATPs. So that is chemiosmosis, oxidative phosphorylation, taking place in the cristae of the mitochondria. So here's a summary. Respiration, brief summary for 1-glucose, C6H12O6. Glycolysis makes ATP through substrate phosphorylation. It is in the cytoplasm. It is anaerobic. It's a breakdown of glucose into 2 pyruvates. Makes 2 ATPs through substrate phosphorylation. Makes 2 NADHs. Follow the green arrow down. They're going to go to the electron transport system, which is going to be in the cristae of the mitochondria. Again, so this does not use oxygen. This is the anaerobic stage of aerobic respiration. Next, the pyruvates are going to go into the matrix of the mitochondria. So the pyruvate is going to get oxidized to a 2-carbon acetyl group. It's going to be attached to coenzyme A. Together, they're going to be called acetyl-CoA. So notice they have 2 carbons. 2 carbons because 2 carbon dioxides are going to be removed. Remember, this does not make any ATP, but it does make electron carriers, which go to electron transport system. Alright, let's see the CoA. Here, red arrow, goes to the Krebs cycle, also known as the citric acid cycle. This is also in the matrix of the mitochondria. Krebs cycle is going to go around 2 times. Cyclic series of oxidation reactions. As a result of going around twice, it's going to make 2 ATPs through substrate phosphorylation, 6 NADHs, 2 FADH2s, these electron carriers, go to electron transport system. 4 carbon dioxide are going to be formed. So now, in aerobic respiration, glucose is completely broken down. Electron transport system, that's the picture we just went through with chemiosmosis. That is in the cristae of the mitochondria. Series of carriers that accept electrons removed from glucose via NADH and FADH2. We're going to pass them along from carrier to carrier, which is why this has the name of cytochrome system, because the carriers are called cytochromes. They're going to make 32 ATPs through oxidative phosphorylation, chemiosmosis, using the electrons and the hydrogens that were dropped off by the electron carriers. Oxygen is the final electron acceptor. Oxygen is going to get reduced, and it's going to make water. CN is cyanide. Cyanide interrupted the electron transport system. So in the electron transport system, the electrons were not able to reach their final destination, which is oxygen. So ATP was not able to be made, and the tarantula died pretty quickly. So usually when you hear the word respiration, you think of breathing. So now we're going to relate breathing to aerobic cellular respiration. Aerobic cellular respiration is just getting energy out of glucose. But breathing is very important because of the 2 gases, oxygen and carbon dioxide. So let's look at this. You breathe in oxygen into your lungs, goes into your blood, carried by your arteries to wherever oxygen needed to go, wherever ATP is going to be made. Drops off the oxygen. Oxygen is the final electron acceptor, so it drops it off. But we also know that carbon dioxide is being produced in respiration. So when we're making ATP, we're giving off carbon dioxide, 2 of them in the transition step, 4 of them in the Krebs cycle. Right? Because aerobic respiration is complete breakdown of glucose. We've got to get rid of that carbon dioxide. That carbon dioxide can change the pH of our blood. We have to have constant pH. We cannot change the pH of our blood. We know all the proteins have optimal pHs, etc. So we got to get rid of the carbon dioxide. Arteries drop off the oxygen, ATP is made, carbon dioxide is released. The veins now are going to carry it back to your lungs, and you're going to breathe it out. Breathe in oxygen, goes to your arteries, goes to wherever ATP is being made, receives the electrons at the very last step of the electron transport system. The glucose is converted into the ATPs. The process. Carbon dioxide is going to release when the glucose is being broken down. Veins are going to pick that up, bring it to your lungs, and you're going to breathe it out. So they're very, 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 very related. So we have gone through 3 types of phosphorylation. Remember, phosphorylation just means making ATP. Substrate phosphorylation, when you're taking a phosphate off of a molecule, in this case off of glucose, that phosphate is going to get put on ADP to make ATP. Oxidative phosphorylation, dealing with electron transfer, redox, chemiosmosis. Photophosphorylation is in flat reactions, making ATP using light energy. So let's go back to this picture. So we followed the arrow down from glucose all the way down to oxygen being reduced into water. So glucose, glycolysis, splitting of the glucose into 2 pyruvates, making 2 ATPs, electron carriers. This is anaerobic in the cytosol, cytoplasm. Pyruvates now are going to be entering into the mitochondria. The transition step or the transition molecule. And the pyruvates give off 2 carbon dioxides, join with coenzyme A, become acetyl-CoA. In that process, no ATP is made, but it does make electron carriers. Coenzyme A goes into the Krebs cycle, which goes around twice. It's also in the matrix of the mitochondria. Here, the glucose is completely broken down by the release of 4 carbon dioxides. The process going around twice makes 2 ATPs through substrate phosphorylation. There's a whole bunch of electron carriers. All the electron carriers go to the electron transport system. Chemiosmosis takes place, makes 32 ATPs through oxidative phosphorylation. And the electrons are kicked out by oxygen, which gets reduced and becomes water. So we're going to have 4 ATPs made through substrate phosphorylation, 2 in glycolysis, 2 in the Krebs cycle, 32 ATPs in the electron transport system through oxidative phosphorylation, with a total of 36 ATPs for 1 glucose. Okay, that's aerobic. Complete breakdown of glucose, so go ahead in blue here. There's glucose, C6H12O6. Glucose has 6 carbons. It's released as 6 carbons in 6 carbon dioxides. Now we're going to go this way. Okay, so let's look over here. Your cells can break glucose down in 2 different ways. One, with oxygen. That's called aerobic respiration. And 2, without oxygen. That's called anaerobic. Respiration or fermentation. Respiration. And different organisms are going to do this anaerobic respiration different ways. So let's look over here. Let's look at alcoholic fermentation. So aerobic respiration. Here's our formula. Glucose, oxygen, reactants, carbon dioxide that is going to get oxidized, carbon dioxide. Oxygen is going to get reduced when it receives those last electrons and hydrogen, it becomes water. and it's going to give off a lot of energy. The amount of energy it's going to give off is approximately 36 ATP. And it's also going to give off some heat. Right? And if you're walking slowly, just doing normal things, you're probably going to be having enough oxygen, and you're going to go aerobic respiration. But if anybody ever has gone running, it's supposed to be going fast. Right? You're going really fast. Probably have enough glucose, but you might not have enough oxygen coming in from outside, because the oxygen's coming in from the atmosphere. So you're going to start to breathe more shallow and faster. And the reason why you're going faster and more shallow is because your body is trying to get oxygen so that you can make ATP. Remember, oxygen is the final electron acceptor. And you're running, and your cells are like, I need more oxygen! Give me more oxygen! I don't have enough, give me more! More! Give me oxygen! I need oxygen! It's getting really desperate, I need it! They're not getting any more oxygen. So what it's going to do, it's going to start to break down glucose without oxygen. Like in our experiment in lab, we had the yeast. We gave it some food. And we were testing to see what kind of food it liked best. So here we gave it some food, C6, 8 to 12, 6. And bacteria just do anaerobic. And look over here, alcohol fermentation is going to produce some carbon dioxide. I'm not going to get a balanced equation here. Plus ethanol, or alcohol, which you can drink. So if you recall, your assignment was that if you had never made any bread, that you were supposed to make bread. And now you have time to make some bread. So you get your flour, and you get your fungus, your organism, right? Yeast is alive. So you get that, get some food, some sugar. And then when the yeast begins to eat the food, breaks it down, and begins to make carbon dioxide. And that carbon dioxide is what is causing your bread to rise. It's rising when you heat it up, it expands even more. And then it actually comes out of the bread, escapes from the dough, and that's where you can have all those air spaces. So it only going to give off 2 ATPs. There we go. 2 ATPs. That is what's going to make the bread all fluffy. So fermentation or anaerobic respiration is just glycolysis, followed by reduction of pyruvate, NADH, to either lactate. Many bacteria are going to use the lactic acid fermentation. That's how cheese is made, that's how yogurt is made. create sore muscles. I've got another type, anaerobic fermentation here is alcohol, like yeast, plant cells, causes bread that rises, leavened bread, wine, beer, is anaerobic. It only makes 2 ATPs. It is incomplete breakdown of glucose, but it goes a lot faster. It's just glycolysis followed by one extra step. Alright, let's look at oxygen debt. When I was little, in grade school, I really liked to read. And sometimes I would pretend like I was sick. One time I drank some really hot tea, put the thermometer in my mouth, and it was so hot I actually broke it. So that one didn't work. So then sometimes I would just purposely miss the bus because I wanted to stay home and read my book. And then my mom said, if you miss the bus, you have to call up one of my friends and ask one of my friends to take you to school. Now I certainly didn't want to do that. So I tried really hard not to miss the bus. But one day I missed the bus. It was an accident. I did not try to miss the bus. So I did not want to call my friends to ask to take me to school. So I knew that the bus stopped at another neighborhood. And I ran so fast. You probably couldn't even see me. I was running so fast. And I made it just when the bus was going to pull out. And I sat in the bus. And I'm going, I'm making all these horrible noises like that. I sat in the bus. I didn't know it, but I had gone into oxygen debt. So really deep, heavy breathing. And my body was restoring what needed the oxygen. So that deep breathing was bringing more oxygen. It was restoring ATP to its former level. It was converting the lactate back into the pyruvate just by doing that. And it works. I was not sore at all. I had to run really fast. I wish I could have stood up and explained, I'm just in oxygen debt. I didn't know. I was totally red. It was a little embarrassing at the time, but now I think it's really funny. This is just a picture to show you that it is just glycolysis, all this is glycolysis, plus one extra step. That's all. You don't need to know all the details. It only makes 2 ATPs. Look at this note over here. Lactate builds up. pH changes, causing fatigue and soreness or burning in your muscles. Eventually, lactate can be converted back to pyruvate by removal of hydrogen and electrons. And then your muscles will no longer be sore. They're actually studying that and they're learning a lot more about that. So they may be changing their ideas on that. Here's a nice chart that you might want to fill out. Make yourself charts like that to study. Here are the answers. Just leave it up for a little bit for you to look at. We actually use fermentation for a lot of different things. Swiss cheese, that's if you start out with this particular bacteria. Carbon dioxide is actually making those holes in the Swiss cheese. Lactobacillus, these different things. Cheddar cheese, yogurt, soy sauce. This fungus here is used for making wine or beer. Striddium makes acetone, fingernail polish remover, rubbing alcohol. That's through fermentation. Vinegar. Alright, that's through fermentation. So we use fermentation a lot for a lot of different things. So depending what you start out with and what you feed it, you're going to get different products. Now let's take a look at this and see how cancer actually is related to the different types of respiration. So the basic difference between normal cells and cancer cells is that the normal cells require oxygen. So the normal cells are aerobic. So they require oxygen to combine with glucose. The cancer cells are anaerobic. So they break down glucose without oxygen, yielding only 1/15 the energy per glucose molecule that a normal cell produces. And this is why cancer cells have a huge appetite for sugar, so not making as much ATP. It's also why people who consume excessive quantities of sugar tend to get cancer more often than those who don't eat a lot of sugar. Anaerobic breakdown of glucose by cancer cells forms large amounts of lactic acid. And the waste product, the same substance formed by fermentation of lactose is in spoiled milk. The liver converts some of these bacteria into glucose in an attempt to salvage a food source from toxic waste. In doing this, the liver only uses 1/5 the energy per glucose molecule that a normal cell can then derive from it. That's 3 times the energy a cancer cell will get from it. So, 3 times the energy a cancer cell will get from it. The more the weak, deranged cancer cells multiply, the more energy is lost to the normal cells. So low levels of both oxygen and energy tend to occur where cancer is present and vice versa. So its wasteful metabolism becomes self-sustaining and dominant unless they increase oxygen and eliminate sugar from your diet. Cancer growths contract and disappear when oxygen saturation is sufficiently increased. Why? Because they are anaerobic. So hyperoxygenating them. They can destroy a lot of things actually. Now we're going to look at something called the metabolic pool. It's going to be what it says here. Degradative reactions, collectively called catabolism, break down molecules and tend to be exergonic. Synthetic reactions, collectively called anabolism, tend to be endergonic. Is it correct to say then that catabolism drives anabolism? Catabolism. We already know that glucose is broken down during cellular respiration. However, other molecules can also undergo catabolism. When a fat molecule is used as an energy source, it breaks down to glycerol and 3 fatty acids, its monomers. As this figure shows, glycerol is going to be converted into pGal, T3P, a metabolite in glycolysis. The fatty acids are converted to acetyl-CoA, transition molecule. The acetyl groups enter the Krebs cycle. An 18-carbon fatty acid results in the 9 acetyl-CoA molecules. In the human body, oxidation of these acetyl-CoA molecules can produce a total of 216 ATP molecules. For this reason, fats are an efficient form of stored energy. There are, after all, 3 long fatty acid chains per fat molecule. The carbon skeleton of amino acids can also be broken down. In humans, the carbon skeleton is produced in the liver when an amino acid undergoes deamination, or the removal of the amino group. The amino group becomes ammonia, NH3, which enters the urea cycle and becomes part of urea, the primary excretory product of humans, urine. Just where the carbon skeleton begins, degradation is dependent on the length of the R group, since this determines the number of carbons left after deamination. Anabolism. We have already mentioned that the ATP produced during catabolism drives anabolism. But there is another way catabolism is related to anabolism. The substrates making up the pathways in this figure can be used as starting materials for synthetic reactions. In other words, compounds that enter the pathways are oxidized to substrates that can be used for biosynthesis. This is the cell's metabolic pool, in which one type of molecule can be converted to another. In this way, carbohydrate intake can result in the formation of fat. pGal can be converted to glycerol, and acetyl groups can be joined to form fatty acids. Fat synthesis follows. This explains why you gain weight from eating too much candy and cake. Some metabolites of the Krebs cycle can be converted to amino acids through transamination, the transfer of an amino group to an organic acid, forming a different amino acid. Plants are able to synthesize all of the amino acids they need. Animals, however, people, lack some of the enzymes necessary for synthesis of all amino acids. Adult humans, for example, can synthesize 11 of the common amino acids, but they cannot synthesize the other 9. The amino acids that cannot be synthesized must be supplied by their diet. They are called the essential amino acids. The non-essential amino acids can be synthesized. It is quite possible for animals to suffer from protein deficiency if their diet does not contain adequate quantities of all the essential amino acids. Okay, so let's go ahead and look at this picture. So this is the metabolic pool concept. When they're used as energy sources, carbohydrates, fats, proteins enter degradative pathways at specific points. Catabolism produces metabolites that can also be used for anabolism of other compounds. So let's go ahead and look at the picture. So remember glycogen from test 1, a glucose polymer. Glycogen is our temporary energy storage in our liver. We need some ATP. We can break one of those glucoses off, and that's going to go follow the pathway that we just learned. But look at fats. Fats can be broken down into their monomers. Glycerol, fatty acids. Glycerol can enter this pathway right here at pGal, right here, right before pyruvate. That can be converted into pyruvate. Fatty acid part can actually go into the transition step right here, forming acetyl-CoA. Proteins, broken down into their monomers, the amino acids. Deamination can take place. Remember, that is one of the amine groups one of the groups that you had to memorize, the functional groups. And then that amino acid can enter here or go directly into the Krebs cycle. So if you remember from our first test, glucose is the #1 molecule used for energy, splitting of the glucose. Remember, fat was the second molecule of choice for energy. That's glycolysis. This is lipolysis, lipolysis, splitting of lipids, fats. Just looked at glycerol here, fatty acids. Remember, fats are permanent energy stores. They're stored without water, so they're very concentrated. There's the proteins. Actually, the proteins that we take in, only about 10 to 15% of all the ATP we make is from the proteins. So sugars, fats, and proteins can all be used in aerobic respiration to make ATP. Again, because the fats are stored without water, they can give twice the amount of energy. Twice the amount of ATP can be produced from the fats. Alright, so this is catabolism. Now let's look at anabolism. So anabolism is building up. So if you look over here what I have on the side, pGal can actually be used to make not just sugars, or 2 pGals to make 1 glucose, but pGal can actually also be used to make lipids and proteins as well. Proteins, the next right here, cannot be converted into carbohydrates. Carbohydrates cannot be directly converted into proteins. However, anything you take in excess will turn into fat. So there really is no such thing as a fat-free diet. Not like, yeah, I eat a whole box of ice cream bars every day, but it says fat-free. I don't know why I'm getting so fat. Because everything in excess gets converted into fat. So that is anabolism. So let's review one more time. Alright, we can break down glucose 2 different ways. With oxygen or without oxygen. With oxygen, aerobic respiration. Without oxygen, anaerobic or fermentation. Aerobic involves all these steps that you see right here. Anaerobic or fermentation only involve this first part, glycolysis, and one extra step. Goes by fast, doesn't need oxygen, but it only makes 2 ATPs. We only do that, making pyruvic acid. We only do that when we don't have enough oxygen and your cells are like, give me oxygen, I need oxygen. It's not getting oxygen, it will go anaerobic. That pyruvic acid can actually be converted back and be reused. I mean lactic acid. I didn't mean pyruvic acid. So when we're not getting enough oxygen, we're going to be making lactic acid. And that's going to be in our bottom picture. Okay, so let's go through here. Glucose. Lysis splitting of glucose makes 2 pyruvic acids. Let's follow that blue arrow here. Notice, look right here. 2 ATPs produced directly. That means through substrate phosphorylation. Does not have to go through any electron carriers. It produces 2 NADHs. Let's take electrons and the hydrogens and the energy over to the electron transport system. 2 times 3 is 6, so it makes 6. So it uses 2. Alright, this is all in the cytoplasm. This pyruvic acid, alright, now is going to enter into the matrix. Give off some carbon dioxide to become a 2-carbon molecule. In the process, 2 NADHs. Goes to electron transport system. Those NADHs drop off the electrons and hydrogens. 6 ATPs are made. So electron transport and chemiosmosis. In this transition stage, right here, the acetyl-CoA going into the right here. This is right here. That's a transition step. No ATPs are produced directly. Acetyl-CoA remaining in the matrix goes to the Krebs cycle. Krebs cycle goes around twice. In the process, glucose is completely broken down. 4 carbon dioxide is given off. Remember 2 up here in the transition stage. It also makes 6 NADHs and 2 FADH2s. 6 times 3 is 18. So it makes 18 ATPs, so electron transport and chemiosmosis. Whereas this one, remember FADH2 comes into electron transport system at the second molecule. So it only makes 4 ATPs. This 38 is if it's a prokaryote and it does not use 2 ATPs right up here. But eukaryotes have to use 2 ATPs right here. I call it the mitochondrial access tool. And these numbers right here, that's basically if it's very healthy, under optimal conditions. So let's review our anaerobic, without oxygen, glucose, becoming 2 pyruvates or 2 pyruvic acids. So it goes through glycolysis. Okay, glycolysis. It only makes 2 ATPs. Well, it actually has a net gain of 2 ATPs. It uses 2, makes 4 but uses 2. Yeast fermentation or alcoholic fermentation goes this way, like with the fungus, plants. That's how alcohol is made. That's how bread can rise. How you get holes in the Swiss cheese. Lactic acid, bacteria, that's how they make kefir, yogurt, different kind of cheeses. That would build up in our muscles. Right when we don't have enough oxygen and we go anaerobic. So all this is in the cytosol or cytoplasm. If it is aerobic, pyruvate acid leaves the cytosol, cytoplasm, goes to the mitochondria, and all that in the picture above takes place right here. 4 carbon dioxide given off. 2 carbon dioxide is given off in the transition stage. 4 in the Krebs cycle. Oxygen is the final lactate acceptor and becomes water. So let's review chemiosmosis. So here's the cristae, the membrane, the inner membrane. Here's the matrix. Here is the intermembrane space. So both the chloroplast and the mitochondria have 2 membranes. So this is the space in between the 2 membranes. So here's our electron carriers. There's NADH. NADH drops its electrons. Electrons go to electron transport system. Here's the hydrogens. So as these electrons go down the electron transport system, energy is used to pump hydrogens. See, these hydrogens are going to get pumped. These pumps pump to the other side. Pumping hydrogens inside the intermembrane space.